Okay,
Okay.
then I think we can start. It is a pleasure to have you, Jordi, presenting today. So I'm just going to introduce you to our audience. We
Thank you.
have currently 26 people online, but I think it will still fill up over the next few minutes. So, Jordi Vives Batlle holds a bachelor's and a master's in physics from the Autonomous University of Barcelona and a PhD in marine ecology from University College Dublin. He became full professor of environmental and radiation physics in 2009 at Westlakes, University of Central Lancashire in the UK, and is recognized as a chartered radiation professional by the UK SRP since 2008. He has 38 years of experience in radioecology, radiation protection, and impact assessment. He has worked globally in Spain, Ireland, the US, UK, and Belgium. And since 2010, he is a senior researcher at SCKSN in Belgium, focusing on the fate, transport, and biological uptake of radionuclides in the environment, and conducting radiological impact assessments to wildlife for industry and regulators. Jordi is a member of the Belgian delegation of ANSCIA, where his activities help develop the international system of radiation protection of the environment, and his current research focuses on ecological modeling of radiation and pollutants transfer and the effects of wildlife populations and ecosystems, with the most recent projects being on modeling radiation or chemical pollutant mixtures and their effects on non-human biota. So, Jordi, once again, it's a pleasure to have you. With that, I would like to give you the floor and ask you to proceed.
Yes, thank you for the honor of inviting me here to present this talk, which will be about a model that I've developed to try to solve the problem of the combined effects of ionizing radiation and chemical pollutants with the focus on non-human biota, the wildlife. Right. Okay, so first of all, I put this in frame of norm sites. And this is for a variety of reasons. Norm sites tend to have some low levels of radionuclides, usually uranium series, but they tend to be mixed with a much more important amount of chemical pollutants. For example, think about phosphate industry, kind of HIPs and situations like this. And their regulators have a problem because they need a consistent approach for impact studies at these sites for the biota. They are worried about how can we compare toxicity and radiotoxicity as the population level for the biota in those sites. They are worried about how consistent the approach of ecotoxicology is with the radioecological approach in terms of assessments. And can we adapt modeling tools from the radiological assessment to include non-radioactive pollutants? This, by the way, picture here gives you an idea of the non-radioactive contaminants that come out from different types of industries, by the way. So the key scientific questions is how the toxicological processes can be combined with the ecological factors, and how can we model the interaction of a concentration of a chemical with the radiation dose of a radionuclide without resorting to complex molecular type of endpoint studies which are scientific studies, but that they don't give the kind of answer that the regulators sometimes want to obtain in a site like this in terms of survival of a population. So to solve this is a norm site we have in Kepkensberg here in the vicinity of SEK. We think about how can population modeling help, because I chose the population modeling as the tool to do this because we're interested in the dynamic issue. In other words, not what's toxic now and what risk is now from what it is now, but over a period of time with a population which is growing and reproducing and passing from one generation to the next, what is the dynamic effect in long term, particularly for low exposures. So we want to do this because we want to consider the effects of historical doses in old legacy sites. Also, the potential possibility that some of the biota can become acclimatized or even adapted to radiation effects. Find out what we need to do to adapt population models, which are a branch of ecological modeling, which is quite well developed, but to adapt it to account for the effect of chemicals on populations. And the ultimate thing is we don't want to change the system of radiological protection and the benchmarks that are already established and that they are, we think, fit for purpose. But we want to change the robustness in situations like this. Are these assumptions that coming to benchmarks like the ICRP, the CRL benchmarks, for example, still conservative when there are chemical pollutants around? And in all, this is done through a mathematical exploration of this multiple stresses problem in population modeling, including temporal scales. And this is the task that I set myself up to do. And this is all based on a key concept that I will show in a moment. I was just to say, most effect studies are at the molecular level with little population studies in the field, which is actually the problem that concerns us in this talk. A relatively simple system of equations that could capture this phenomena in a simplified way would be useful to provide guidance to regulators. This is done by this model here, which I call the repair dynamic sort of repair and fecundity dynamic model. You have to imagine a population of non-human biota as being composed of healthy individuals, which are called X in blue, and sick individuals, and that the healthy can become sick, but that the animals, so let's say animals, or it applies to plants too, have a repairing capacity, which we call the repair pool, that is capable of returning some of the sick back to healthy. There is also the fecundity that allows these things to reproduce, and both the healthy and the sick can reproduce. But if this repairing pool and fecundity pool can be put mathematically as a function of doses and concentrations, then you have a model that regulates what is healthy, what is sick, what is being repaired, and even what could be potentially adapted or acclimatized to radiation at very low doses. And there is a mathematical system of equations that I found out that is capable of working under that scheme in which reproduction and repair are sources of energy that are depleted by the presence of the contaminants. And I will show you that this can be developed and coupled to ecological processes, thus developing a relatively simple population model. This is for radiation alone. We did already some years ago. We did that as part of Project Modaria. I'm not going to describe maths here. I'm just going to show them because, but I'm going to give some very basic things into it. But in this case, you have a system of 4 equations in which you have healthy, and the affected individuals, the recovery function, and the fecundity function. And without going into mathematical details, we apply what is called the logistic model. That basically means that the rate of change of a population is proportional to the reproduction rate and to a term that becomes smaller as the population reaches a certain limit to account for the inability of an ecosystem to host more than a certain number of individuals. And if then you have a subtraction term in here that depends on the dose, and then a repairing term that sends some of the sick back to the healthy, as you can see here, you can in principle set this up and see how good it works in a real situation. Now, luckily, the ERICA tool, that is the tool for non-human biota impact assessment, has a database which is called Frederica. And this database contains a review of the available experimental data in the following format. For example, at a dose of X, how many are killed at various levels of dose? Or at a dose of Y, how many become sick, or what is called the morbidity? And then if you put in the x-axis the experimental doses at which, for example, mortality occurs, and then in the y-axis what this model predicts, you should obtain a line y equals x if the model is set up properly. And this is what we did. We observed that within the uncertainties associated with these types of measurements, we could have a validation of this type of repair, logistic, differential equations system to the data that is in the ERICA tool. without having to look at the molecular effects, just by defining this repairing pool as a function which can be depleted with a certain shape, and we collaborate and parameterized with some data that we had, you can get this. So we can have something relatively simple without going to the DNA level and starting looking at all the genetics effects that would really be impractical for a model that is designed for regulation. Now, the key is to apply this to the, and this is the last math you will see, by the way, to, to chemicals. And basically, it's an extension of the model that, that I just showed. You have healthy, sick, the W, which is the organisms that can become adapted, which is a thing we were testing that we don't always use, the fecundity pool and the recovery pool. And this equation has a series of terms. The green ones are an auto-recovery, meaning that whatever you subtract from these equations, the model will tend to auto-recover on these functions to a limit that is K, which is the so-called the carrying capacity of the system. So the green stuff is the basic population model that is well known in population ecology without anything else. The gray stuff that you can see in here, I don't know if you can see the mouse moving. Can you see the cursor when I do
Yes,
this?
we do see it.
Great. Because these gray terms are the death, the natural death, the death that occurs as a result of sickness and predation that comes from ecological data, from ecological databases. So that has to detract from the tendency to recover. Then you have the blue terms, and this is the recovery. So if the sick is the y, that little quantity kyr becomes negative here and partially goes to healthy and partially goes to adapted. So this is to account for the recovery. And in the fecundity in the recovery pool, they also have similar equations. And the important thing is the yellow part, because the yellow part tells you how the contaminant and the radiation act. See a function alpha, which is a certain constant, multiplied by radiation dose in milligrams per hour for a biota, plus beta multiplied by the concentration of the chemical in milligrams per kilo, okay? And these terms are additive. And then there is another term that is a cross product of these two. And I will tell it later, but I add this term because the effects sometimes don't work just on addition, but there is an extra addition or detriment, which we call antagonism or synergism, to account for the toxicity, okay? And this is the basic, the basic layout. And you don't have to remember this, but just have to believe that the system of mathematical equations with 6 equations can capture in a phenomenological way, which we mean effective empirical way, this combined effect of the radiation and the toxic. And this ends the maths lecture part of the of this talk. This I set up in a software called Model Maker, and this is just to show you how it looks. This is a very typical type of software that shows you, that is very visual oriented. So you can put your boxes, and then you have arrows to go to one compartment to another. And if you click on these arrows, you can put your equations in there. And then there are influences and variables to calculate certain things. And then this double box here shows a very long list of parameters that is included in here. So it's just to say it's no longer necessary to take a code and put it on a code. It is easier to program on a visual interface because you don't do mathematical errors when you let the system do the integration and the calculations. And also you can extract from Model Maker a script which contains the code that magically is generated once you've entered all these compartments, and you can pass it to a colleague that is programming this in Python or Fortran or something else as part of a bigger model. So it's a very convenient way to work. The question is how we compare directly the effect of radiation and a chemical, because radiation is in milligrams per hour and the chemical is in milligrams per kilo. And how can we compare these two quantities? And the trick is in these coefficients that multiply alpha for the dose and beta for the concentration. And basically, these maths are basically here to say that because the activity is the number of atoms that are decaying multiplied by the decay half-life. That's the equation, sorry, by the decay constant, activity equals n lambda. You can convert those from becquerels, sorry, the activity from becquerels per kilo to how many milligrams of radionuclide are there in that kilo to give that amount of becquerels. This is a simple conversion from a concentration to activity. And then by multiplying by those coefficients that are that convert from becquerels to those, you can develop a formula that links those of a radionuclide in the organism to the concentration and in terms of moles, milligrams per kilo of that element that is radioactive in that organism. So this is what I call toxicity doses or toxicity units. You can express those mathematically as a concentration of some mysterious chemical that has the effect of through the radiation. You see what I mean? And once you have everything in the same units, thanks to this equation, you can see if the effect of the two, the dose and the chemical concentration, is either simple addition or any other type of effect, combination effect. Because in toxicity, you have two models. One is concentration addition, which is when two contaminants have the same mode of action, and therefore you can treat them as dilutions of each other, and this is the concentration addition. Or independent action, which is that they act independently. And here we make the hypothesis that the radiation and the chemical affect the repair system in the same way. And this is a reasonable hypothesis in light of the experimental data that we've found on experiments where they've added chemicals and radiation in the same animal and try to find a combined effect. And that saves a lot of effort. This model, like any other, requires parameterizations, and although I show the equations here so that when you look at, if you want to copy it or have a look at it afterwards when you get the recorded lecture, suffices to say that the key parameter for the radiation part of the model is the LD50, which is sorry? Did I hear something? Can you hear me?
I don't think so. It's all good. You can go ahead.
Okay,
We
okay.
can hear you.
The LD50, which is the dose you need to give to the animal to kill, to the population to kill 50% in the absence of other ecological factors. This is a very known quantity in radioecology. And in the chemical world, there is also the LD50 and the EC50, but in this case, it's not the dose you need to kill 50%, but it's the concentration you need to kill 50%. And because they have different units, there is this 30 number here, which is 30 days, because the definition of LD50 is the dose that you need to kill 50% in 30 days, okay? And the same for the EC50 30s. You can put them on different time scale, but we use 30 days for compatibility with the radiation. And this alpha and beta here are the parameters that convert concentration to those. And this gamma is the one that if it's not zero, you don't have simple concentration addition anymore, but you have that your behavior is deviates, as you can see in this graph here, from concentration addition, detracting or increasing, depending on whether sign of this parameter, leading to the antagonism and the synergism that the toxicologists use. Now, this graph is to say that to kill 50% of the population in a long, by the way, 30 days is not very useful when you look to population ecology, because the population stay there for years. But with the units conversion, you can change 30 days to something more reasonable, like 1,000 days, which is the 3 years, and which is sufficient time to see an effect in many cases on a population. This graph is here to show that in the case of that to give 7.36 milligrams per day to a biota population, it's milligrams per day. You obtain the same effect that if you give 3.98 milligrams per kilo. So 3.99 milligrams per kilo of a chemical will give the same effect as 7.36 milligrams per day of a radiation after 1,000 days in killing 50%. And this dose and this concentration are called the PD population dose 50 in 1,000 days and the population concentration 50 in 1,000 days. This is typical concentration addition behavior because all we've done is a change of units between dose and concentration. Now, the parameterization of this model is a tricky job. On the radiation part of the story, we base ourselves on a series of publications, both by me and colleagues and by some Russian researchers, Sazikin and Khrushchev, that for years have been using this type of model for radiation. And so we build on their data to parameterize this model. There are some theoretical values that you can assign to some of the parameters. For example, the death rate for unrepairable individuals, once the repairing function has gone to zero because the radiation has killed it, should be such that if you administer an LD50, population should die in 30 days. And therefore, it has to be 1 divided by 30 days, that rate. And the same applies to other parameters that they have to do with the damages of the reproductive system and the repairing pool that are obtained on the basis of certain assumptions, such as that that reproduction is more sensitive endpoint than mortality, for example. And this is and then morbidity. There's reproduction first, morbidity second, mortality third, and that poses certain constraints on some of these parameters. There's no time here to go into this, but just to say that for some of them you can deduce reasonable values, and that's it. So what problems do you encounter when you apply a model like this? Everything looks reasonably pretty in terms of we have a closed, nice, elegant system of differential equations, some parameter values, but what is the problems? Because every modeling has problems. And the first problem is that the EC50 for chemicals is the LD50 also, but particularly for chemicals is highly variable quantity. For example, to kill 50%, to affect 50% of the population by adding a toxic chemical depends on the administration route. For example, it's not the same as giving a collection of mice an injection of the toxicant or to give them as a food. Or it was a subcutaneous application. Values can vary significantly. Also, different compounds of the same element can have also variable toxicities. As you know, for example, I think it's the chromium or the cadmium, both dependent on the oxidation state. The speciation, therefore, is important. And also, if you're using organisms in the lab to do experiments like mice, for example, various strains of the same mice can give different results. So the range is quite broad. And it also depends on the specifics of the study. Was it an exposure time in an acute situation where you gave a single high dose, or a slow chronic situation, or a multiple dose intake? And to some extent, it's impossible to disentangle this in a model. So you have to do approximations, because what we are looking here is orders of magnitude of the dose and or the concentration that can affect the population a certain respect, rather than the exact value of the dose. It is possible to scale results of the chemical doses triggering endpoints in our model for different EC50s, because by application of scaling factors to the date, but that's not very recommendable, ecotoxicologists say. So it's just an approximation. And EC50s, as I say, for chemicals have more variability than LD50s due to the ranges, the effects that I mentioned. Anyway, here we made a mixed contaminant test scenario to test the model, because that's the first thing you need to do is to set up some simple test scenario in which you will either test your model, make some predictions, and the difficulty is that there are no field studies. And what we are interested in precisely that: low doses, low chemical concentrations for a very long time, rather than a huge thing. But then it's a matter of comparing models. And in our case, we use the bull, the Microtus agrestis, is a little animal, the field bull, which is present in Europe in many places, and for which we found more data than for other animals that we looked at. And then we can test. The first thing to test is the effect of adding the chemical progressively for the same, for varying doses. So you start with a concentration of chemical of 0, and you do a range of doses. Then you add a bit of the chemical, you repeat, and see how the answers of the model begin to change. Here is a single pollutant in which you have remember that function alpha d plus beta c, the function that detracts from the stability of the solution of the model from the healthy. So this is, instead of varying d or varying c, I vary alpha d or beta c, because then its model is symmetrical between the two things, because it's toxicity units. And here you could say, for example, you vary the model, you start increasing alpha d or beta c, should have the same result. And then you begin to see the population initially has a stable level. You add a bit of contaminant, it begins to reduce, but goes to a slightly lower stable level. You keep adding, and then it keeps reducing. But then, all la la, suddenly, there is one magic number for which the system collapses. This is called the tipping point. And this model is good in reproducing tipping points, which is what the ecologists observe. And remember, I said that there is a mechanism that I put in the model by which you can consider that at very low doses or concentrations, the organism could be adapted as if it became temporarily vaccinated. The correct terminology is acclimatized for a short time, making it somewhat protective, and because in radiobiology they are discussing the potential possibility of beneficial effects of low doses of radiation, and we put that in the model. And you can see, although it's very difficult and this part is entirely theoretical, that it helps to prevent population collapse for certain levels of dose. Then, sorry that one of my graphs seems to have overlap here in the picture, but the idea here was to start varying the dose rates for 0 chemical, 1 milligram of chemical, 2.5 milligram of chemical, 3.5 milligram of chemical, and so on. And the interpretation of these graphs is that the dose for which a model predicts a population collapse or tipping point decreases as you start adding chemical. This is not surprising since the two are acting together on these repairing mechanisms. But this is telling us something actually very important, perhaps the most important thing. If you have a legacy contaminated site where there is, for example, a lot of radium because of some phosphates, and it's all full with phosphates and calcium and all sorts of other contaminants in there, and we see that the biota is being affected, be careful, because it may be that it's not the radiation that's at fault, but the chemical contamination. And this model allows you to put the two together, and so help you predict who is to blame, in other words, for the decline of the population. And this is what regulators need. They need to know how much populations are being affected increasingly when there is chemical, because the sensitivity of the organisms are being increased by the chemical, even if the radiation remains constant. And this is the interpretation of these graphs. This is the kind of things that regulators would need in order to classify a site in terms of the level of pollution, chemical and radiological, based on the parameters that the site has for a model like this. This acclimatization that I explained is a phenomenon that appears at very low doses of radiation, and to cut a long story short, is proportional not to the dose, but to the cumulative dose. In other words, the dose that has been accumulated, integrated. So not to the milligrays per day, but to the milligrays that the organism has accumulated in its body. and that if you start increasing this dose, the acclimation begins to become less effective. In these graphs, you have the blue, which is the healthy, the sick, and the acclimated. And by the first graph in the second row, you can see that the acclimated population is increasing, and is in certain conditions, it can be high enough to prevent what otherwise would be a collapse of the population. And this is what we were trying to work out in these graphs. This is another example of varying chemical concentration on the survivability of the population with the acclimation enabled for various concentrations of the chemical in the system over time. And it's basically telling us the same story. And this is the graph that I like the most. I'm very fond of this graph, even it's a little bit blurry, because we tried to come with an example where I said, aha, all I need to do now is to prove what is the amount of low concentration of chemicals that can acclimatize my population such that it doesn't collapse due to the radiation, whereas without the chemical it would. And I couldn't find it, no matter how high. I could delay the effect, but I couldn't find it. And this got me very puzzled, because I wanted to know if this could be viable. This is a more research part here. And then I realized that because it's an integrated dose that the acclimatization responds to, if you start gaining dose, you'll see acclimation, but then you'll go past that, because the dose will then be accumulated dose will be too high, and the acclimation will go down to zero again. So this is a transient effect. Aha, but what if I start with an accident situation, and that's no longer for a norm site, it's an accidental situation in which there was a very high dose at the beginning, and the population started to crash, and then as the dose went lower, there is a point in which, with and without acclimation, I've reached the point where the cumulative dose at that point is not too high to kill acclimation and not too low to miss it. And this is what we did here. And we find an example in which in the left, you can see as the time increases for this particular dose profile, which we did it for we used the Chernobyl Red Forest dose profile from a previous paper. You have integrated dose that goes up, and then it goes down as the dose decreases. The healthy are the blues. In the left, the blues collapse after a certain point because the dose has accumulated and it begins to collapse the population. In the right, you see that the green dotted line of acclimated organisms increases enough to stop the collapse of the population for that little time where the population would collapse. And after the dose continue to decrease over time because of decay, it has rescued the population. So this is a theoretical, and I stress theoretical, suggestion that the acclimation could be important to sustain a population exposed to a decreasing level of radiation dose. And now, coming to relatively to the end, one more thing. I did an experiment. Because I don't have access to a lab, I do my experiments in the computer. And I did what my colleagues do in the lab. They add a certain dose to plants, and they measure how much have been killed, and then they add a bigger dose, and they see how much have been killed, and they keep making a curve. And this is called the dose response curve. And those who are working on effects, you'll be familiar with this curve. It follows a certain equation called the logistic function. So I started using the model as if it was my lab. Okay, I simulate how much survive after a long period of time, which was 3 years, over 1 year, sorry, after certain doses, and I predicted the mortality. And it gave me the same dose response curve, fitting to the same equation. And because this equation has well-known parameters, is this logistic function that I explained here, which in this particular case have this numerical value, but they vary depending on the experiment, then it was a bit of an eureka moment, because the experimentalists, what they do is dose response curves in the lab. So if I can link my model parameters, alpha, beta, and gamma, to the parameters of this curve, I can validate the model with experimental data using dose response curves. I mean, this is really cool because I can vary my parameters of my model until I obtain the dose response curve that the people have measured in the lab, and then I'll have the model properly set up for that type of organism. And vice versa. I can work both forwards and backwards. So this is a good, and if it deviates from this behavior, I can also say something about the value of the parameter gamma, that is the antagonism and synergism. So I can link this model to the experimental data. And that is, it's as far as I progressed at that point, because to establish the theoretical model, to see how it's parameterized, to understand the behavior of the model on the various different concentrations of chemical and or radiochemical, and to see how it could be linked to the experimental data. Of course, what follows next is to try to apply this in the real life. We published this in the JRP, in CETAC, where it was quite well received. And in fact, the ecotoxicologist there told me that some of the parameters were meant this, or meant concentration addition, or meant independent action or synergism, which I didn't know because I'm just a poor physicist. We did a tech doc for the IAEA on this, on Project Modaria. And now I am working on various areas. One of them is benchmarking, which means if you cannot design a field experiment where for 30 years we give a certain concentration or chemical and dose and follow it over time, because it's not possible to do that now, at least compare with the predictions of different models that are completely different but aim at solving this type of problem. Things like the mixture toxicity potential applied adjustment factor MSPAF model of the French colleagues of IRSN, things like depth modeling, the dynamic energy modeling, things like depth-tox modeling that is derived from depth modeling, things like what's it called? These models that follow populations, individual-based models, to see if they give similar answers. This is like climate change modeling. You cannot predict the climate validate your model for what will be the climate in 1,000 years, but you can compare with lots of other models and have a curve of the outputs of these models to give you confidence. So to try to do something like this. We also, another avenue is comparing with experimental studies. There are studies from the STAR project, EC STAR on mixture toxicity, particularly in plants, studies on Atlantic salmon exposed to heavy metals and contamination from the Norwegian colleagues at Serrat, investigations on the effect of uranium and gamma radiation in Arabidopsis thaliana at SEK, studies on lemna minor plant involved in contaminants, both chemical and radiologicals, by the Aveiro University or SEK. And there are various studies where we can mine to validate the model, because population level studies involving radionuclides are not available. And this is a striking knowledge gap given the situations in which mixture stressors exist. So the conclusion, we have a model and parameters consistent with at least the radiological part is well calibrated and validated with the Frederica database. There are possible validation routes, experimental, I've mentioned some, also the Drosophila, in vitro, lemna minor, zooplankton. From now, these predictions are qualitative due to the uncertainties. Exploring non-linearities in model solutions is something that we should do now. Find situations, it's like the tipping points, find situations where you enter into the chaos dynamic, which is something theoretical. Compare model-generated dose response curves with those from the experiments. Deduce restrictive situations in terms of the effects to the population in mixture exposure situations, like how much maximum of this chemical can you have and this radiation for these sites. Explore this equation, alpha, beta, and gamma model, formula that I have, to link it to concentration addition and independent action. Then use these parameters, alpha, beta, and gamma, as index quantities that could allow regulators to classify pollutant sites, open a dialogue between radiologists, ecotoxicologists, and regulators to see how can we continue to simplify and apply these models. One of the things I've been doing, nearly there now, is to apply this to the Oslo Fjord as part of the project Mireia with the IAEA, and in there I've extended this model to a toxicokinetic, toxicodynamic model, but by adding a transfer part, so no longer dose to the biota or concentration in the biota, but from the environment dynamically to the biota, and then the rest of the model. We are doing this as of now. And eventually, we would like to convert this model into a practical tool. I've mentioned Mireia, the TD toxicodynamic model. To make it practical, we should make it user-friendly, including a proper using interface. We should have indicators to show where the model points towards population effects that should be considered, because it puts compliance with the benchmark perhaps into question, maybe a kind of a traffic light system like the ERICA tool has. Also, the model should point out clearly what the uncertainties are and should show the reliability of the assessment based on quantified uncertainties. And this is all work for the future, probably beyond my career, because I just have 6 years to go. I might have just enough time to do the next part of this. And I thank you all for your attention. Here is a list of references which you will get with the slides or the recorded version. And I just wanted to add one quick word in memory of Professor Nick Beresford, who was the leader of the exposures and effects group of the project Modaria, where I developed this model and provided essential ideas and collaboration for this, and we are missing him indeed very, very much. And for this, I thank you for all your attention, and I'll answer any questions that you have.